Everyone, this is week 5 of the course, and we are having module 5, sequencing part 2. We're going to continue learning about different technologies that are out there trying to read information from our genetic makeup or other species and generate data where we can do many tasks trying to understand the underlying mechanisms and how to better treat some diseases, for example. It is an interesting subtitle here. We're going to look into third generation sequencing. So I hope everything is going all right so far. A good reminder that I want to give for everyone as you watch this lecture, it's really a good time to make sure that you have been in touch with your team. There is the team project. Some communicated with me regarding the choice of the paper. I gave some recommendations. We had a good discussion last office hour. This is also an invitation for many students to try to join if they have any questions. Teamwork and team project is very important for the course. You want to sit down as a team, communicate online, whatever means is good for you, and find a good paper. Find a good paper where you have some resources to reproduce the results and share with us towards the end of the course. Great. So in this part of the course, we're going to learn about next generation sequencing. So we wrapped last week the discussion on Illumina sequencing, SBS sequencing style, and then we looked into Illumina sequencing. We discussed some interesting ways into how we discussed Sanger sequencing. Illumina is something we're going to discuss in this part of the course, okay, which is part of next generation sequencing. We looked into Sanger sequencing in depth and in details, and also we're going to cover some of the recent technologies that are third generation sequencing. In this part of the course, we'll see what's called FASTQ files, actual real raw data files that we get from the sequencing machine with some sequences, with some ACGTs that we can read through. So in this part, you're going to learn about sequencing technologies, including, as I said, next generation sequencing. Key areas in all sequencing strategies, we're looking at what? Fluorophores, right? Something that we can measure in terms of intensity of light, light intensity, some screening mechanism like some laser, right, technology, some flow cells, some wells, some plates, some dishes where we can have some designed probes and see if these DNAs that we're going to wash over this is going to bind, and then based on that we get certain measures. Some key ideas that are common between most of the sequencing technologies. Now, why does it matter for you as a bioinformatician to learn about next generation sequencing and third generation sequencing, all these ideas? Why would this matter? Well, it's part of the bioinformatician's role analyzing sequencing data. So you may expect as a bioinformatician, if you want to work in this area and as a profession, you might be tasked to work with sequencing data. And as a bioinformatician, you are expected to know something about sequencing data, which would mean knowing something about the technology, knowing something about, showing understanding of the underlying technologies and the data that comes from that technology. Each specific technology has some unique features and some unique outputs. So processing, analyzing, and interpreting this data correctly requires understanding the technology. Sometimes you may dive into developing your specialized tools or your own pipeline, depending on the needs of the project, depending on the specifics of the data samples that you have, and so on and so forth. So it's really an important thing for a bioinformatician to know something, and more than something, for about when it comes to sequencing technologies. So we're going to start now with next generation sequencing.